Okay, very good. So I thought what we would do is step back for a second, do that kind of thing that they used to have. Well, I guess they still have them at the malls and stuff like that, where you walk in and you're looking for a store and it says you are here. So I thought we would kind of remind ourselves where we are. When I stopped yesterday, I stopped. Yes. Well, yeah, I stopped just as we finished talking about what was going on with this atmospheric oxygen, right? You remember before that, and that's why it was good that we did the lab yesterday. Remember before that, the world was populated for a long period of time by these dense concentrations, these dense mats of photosynthetic organisms, right? Photosynthesis, of course, you learned in Bio-1, as I said yesterday, produces oxygen, right? So these photosynthetic critters were producing oxygen, and unbeknownst to them, especially the ones, that were anaerobic because oxygen killed them off, unbeknownst to them, this rise of oxygen in the atmosphere led to another expression of an advantageous variation. I always want to kind of remember to step back to our roots in evolution, because I told you at the beginning of the semester that this class was basically a class about evolution. We're going to talk about the diversity of different, plants and animals, but again, diversity and evolution, by now you've realized that this one and the same. You can't talk about one without the other. So I want to remind everyone what was going on during this time, right? The prokaryotes, the photosynthetic ones, they're not all photosynthetic, but the ones that were, especially the oscillatoria and the Anabaena, and Gloeocapsa that we looked at. Remember green ones? I said green usually means, you photosynthesize. They were producing lots, and remember, there were huge stromatolites. So these big mounds of them. There were just tons of these things out there. So they were producing this oxygen. It took a while to rise. I mean, let's see, 2.7. Looks like about 1 billion years if you're kind of going by that real rough scale there. So for about 1 billion years, the prokaryotes had the place to themselves, and they were photosynthesizing, and oxygen levels were rising, and they were getting, higher and higher. And again, without having any goal in mind, evolution never has a goal in mind. This led to a fortuitous advantage, and that fortuitous advantage was critters organisms who could aerobically respire. Remember, now you understand why you got to do the Bio-1 thing before the Bio-2, because you got to understand the significance of what was going on here. Organisms had figured out a way to take glucose, to take sugar, to take food, and get, what do we calculate, 15, 16, 18 times more energy out of it if you did it with oxygen, if you respired with oxygen versus the other kind. Remember, anaerobic, right? There's an anaerobic respiration in which you only get 2, we said, right? So think about that. That's an advantageous variation. So we are here, we are right there as this rise in atmospheric oxygen, is beginning to have another effect, right? So these are building upon one another. The rise in prokaryotes, obviously specifically the photosynthetic ones, led to the rise in oxygen. The rise in oxygen led to organisms that could do more with the amount of energy they were getting, right? 18 times more, 16 times more, so you could be bigger, you could be more complex. Think about getting that extra energy as making more money. Now you can buy more things. I mean, you got a TV and you got a this and you got a that. When you have 18 times or at least 15 times more energy coming from the same glucose, you can be fancier, right? You can afford to have things like organelles, right? So you can be a eukaryote. And now, first you're going to be a single-celled, and they're around for, what does that look like, 750 million years. So they're around quite a while. But the eukaryotes, you remember from Bio-1, again, going back to that, the eukaryotes had some very different characteristics than prokaryotic cells, right? For one thing, the nucleus, the prokaryotes don't have one, eukaryotic cells do, but much more than just the nucleus. Eukaryotes have many chromosomes, prokaryotes have one. So there's this idea of overall genetic complexity, right? So that's in there. And then, of course, these organelles. And organelles allow cells to be very efficient because they can compartmentalize and they can specialize. And you got the respiring organelles over here, the mitochondria. And if you're a photosynthesizer, you got the chloroplasts, and they're photosynthesizing over here. And the endoplasmic reticulum, right? Blast from the past. Isn't that right? The ER is transporting things, and the ribosomes are making proteins. And all of this is happening in a compartmentalized and not the only way to do things. I got to caution you as we go through these discussions about evolution and diversity of organisms, when I talk about the ones that we might think are simpler. Who else is coming in? Hi, Jesse. Good afternoon. Suzanne, how are you? I'm sorry. I saw you guys come in, but I was kind of in the middle of something, right? Right now I just lost track of where I was. Well, what I'm trying to say is, no, what I am saying is, remember that prokaryotic organisms, even though, good afternoon, even though they're what we would call simple, they're still pretty efficient. At least the ones around today are. They've been around since, or at least their ancestors have been around since the beginning of time, and they're still around. So when we start talking about the mosses and these really simple critters like worms and jellyfish, don't think that we're talking, don't get Aristotelian on me, right? Don't get Aristotelian on me. Don't start talking about ladders. Remember that these critters that are around today, these organisms, can be relatively simple, like the prokaryotes and the mosses and the jellyfish, but they do really well in what they need to do to adapt. Back to old Chuck, we can never forget Charles Darwin, right? So what is this whole idea of eukaryotic life? Now, there were many things that were the result of this rise of eukaryotic life, just like the rise of eukaryotic life was the result of oxygen levels rising, as I've been describing to you. Now, the anaerobic prokaryotes that I mentioned the other day, we'll talk about those in a second. All right, so we go back and we start looking, where did the eukaryotes come on the scene? Well, again, hard things to fossilize. 1.8 billion years ago, possibly, based on looking at things like a genetic clock, of the mutations that were necessary to get something eukaryotic. Oldest known confirmed, I believe, the book said, and I looked online, I didn't see anything different, was, give or take a few hundred million years, 1.2 billion years ago, right? So even the eukaryotes, even the multicellular eukaryotes are starting to rise pretty quickly. So we went from single-celled eukaryotes. Let's see what the back here when it comes to, where's our little, oh wait, our map. Hold on a second. Where's that map? Yeah, well, they have it 750,000 years before the multicellular eukaryotes. But nonetheless, once we get to that point, so I guess about 1.8 billion years ago, we start to see we are. But what was it about eukaryotes? Let's talk about what they could do, how they could gain an advantage, how they could rise in prominence. I just got something about a bad connection, so I'm sorry to. Can everyone hear me? It's telling me my mic is. All right. Can everyone hear me now? I just got it. Yeah. Okay. All right. Very good. I'm sorry. I just got some sort of a thing told that my connection was dropping. All right. Sorry about that. Anyway, back to our eukaryotes, back to the star of the show at this particular point, right? What's going on is these multicellular eukaryotes begin to evolve. The idea of being a eukaryote means a lot of things in terms of complexity, like I've been talking about. But, and the development of organelles, which prokaryotes don't have. And organelles are the parts, again, the mitochondria. So you got this division of labor. If you want to think about it in terms of analogy, think about it in terms of in animals, how as we observe more and more complex organisms, animals, we see things like tissues. We see like there's heart tissue and liver tissue, and there's an organ. So there's this division of labor. So kind of think of that in terms of what's going on in the cells. And that's allowed these cells to be efficient and to be able to exploit different environments. Now, another thing about prokaryotic cells that the eukaryotic cells had over them was eukaryotic cells had a cytoskeleton. Now, I don't know if you remember, there were three parts. the microtubules, filaments, the actin filaments, right? There's another name for one of those, microtubules. Anyway, these parts of the cytoskeleton were things that gave structure to the cell, but they had a variety of jobs. You remember, even in mitochondria, the spindle fibers were made up of microtubules. They were made of parts of that cytoskeleton. One of the other things that cytoskeleton allowed eukaryotic cells to do that prokaryotic cells didn't have the ability to do was engulf things. See the picture down below? I'm reminding you this idea of endocytosis. So eukaryotic cells began or achieved, developed the ability to engulf things. Now you're thinking, okay, engulf food, right? Engulf food. So they could go out there and actually engulf things. And one of the things they might be able to engulf, and this is where we go back, this is really kind of cool, because we go back to the beginning of the arms race and who eats who. Maybe these eukaryotic cells, especially the single-celled eukaryotic ones, learned how to engulf the bacterial cells, and even learned how to engulf the photosynthetic bacterial cells. So I don't got to photosynthesize. I can just eat these other things that do, or I can eat these other things that eat these other things. So right there at the beginning, oh man, isn't that cool? Right there at the beginning, we start to see this evolution of predation. Predation is one of the strongest driving pressures in evolution. So let's go ahead and go with that. Let's say eukaryotic cells develop the ability to prey upon other things, among other things in endocytosis. It's not just food you're eating in endocytosis. Sometimes you engulf other things. Like if you remember, what was it, receptor-mediated endocytosis, in which specialized thing. A second as we go on my mouse. There we go. Click. All right. Now, so here's another thing that what happened. We could assume back in that time period. There we go. So going up to the top is this, whatever this ancestral prokaryote / eukaryote, whatever this prokaryotic cell that developed the ability to engulf, and that's what you're seeing in the next picture. So the way engulfing works is you would just basically fold in on yourself. So the purple that you see there is coming from the outside of the cell in. So the membrane of the cell developed this ability to engulf. That's how you would endocytosis. You would fold. You either bring it in is kind of one way of looking at it, or you fold around it is another way of looking at it, okay? So this, and again, where does that go on the ladder there, Aristotle, right? Prokaryote, eukaryote, but somewhere along the line, some prokaryote developed or gained this ability to engulf things through random mutation, right? And so we kind of run with that for a second, and let's follow the set of cells. Let's follow in the order that we got there. So one of the first things that would have happened, and one of the things that would have been probably favored, remember, we always have to see things that persist as something that gained or derived, what is the word I'm looking for? Imparted, that's not even a word, or gave an organism an advantage, all right? Gave a living thing an advantage. Now, what's the advantage here? Wrapping up the nucleus. See, those pinching in of the cells could eventually form membranes, right? That's basically what all cell membranes are made of, the same thing. The ones on the outside and the ones on the inside are all phospholipids, right? So the membranes are wrapped around the nucleus, and that would kind of protect the nucleus where the chromosome is, where the genetic material is. You don't want to get that messed up. And parts of it could become the transporting part, the endoplasmic reticulum. Remember, that's how the messenger RNA, by the way, gets out into the cell. Clever, huh? All right, but what if these engulfing of cells were engulfed something like, say, an aerobic bacterium? Now, okay, in the lower left, I have a picture of one that's fossilized, been fossilized in amber, right, where they find like mosquitoes sometimes. This is a cyanobacterium, and actually, no, I'm sorry, that's not cyanobacterium. I should not say that. This is a proteobacterium. My goodness, there's a mistake. I'm having a semester where I find all the. I've been using this slide for quite a while too. That's supposed to say a fossilized proteobacteria. Give me 1 second while I go into my original slide and change that. I thought that looked funny because if it was a cytobacteria, it would be a photosynthesizer, and it's not a cytobacteria. It's a proteobacteria. Proteobacteria is a heterotrophic bacteria. It does not photosynthesize. So, just erase the cyano there, or actually just the cyan, and put proteo. Anyway, here's what I'm trying to get at. That proteobacteria was an aerobic bacteria, and a cell engulfed it, maybe with the intent on eating it. Now, for whatever reason, the other day, a symbiotic relationship, you help me and I'll help you. All right? Not always help. Sometimes like predator-prey is symbiotic, but the symbiotic relationship here would have been that the aerobic bacterium gains a protection and a place to live. This, inside of another cell. So instead of being, kind of out there where other cells could engulf it and eat it, it's sort of inside the protective cocoon or layer, or membrane is the right term, obviously, for where that aerobic bacterium is. And that means that cell is very good at engulfing, could also be very good at engulfing food. And so the bacterium says, well, you're really good at gathering resources to the cell, and the cell says, yeah, you're really good at cranking out ATP really efficiently with that oxygen, so how about we do this together, right? How about we work this out together? Because now think about it. Step back for just a moment. Before a cell had an aerobic bacterium, before the aerobic bacterium was around, and if you follow the arrow now down to the left, the aerobic bacterium through time becomes the mitochondria. That cell was limited to only anaerobic respiration. So back to your Bio 1 again. Remember, we don't start getting oxygen involved until we're in the mitochondria. The anaerobic, the fermentation, the one where you only get two, happens in the cytoplasm. Everybody's got a cytoplasm, even if you're a prokaryote. So this aerobic bacterium, right, was doing its thing and learned how to actually use oxygen, or develop the ability to use oxygen to get energy. And so this symbiotic relationship occurred, right? You help me and I'll help you. Now, likewise, those cells that eventually ended up becoming those parts of photosynthetic critters would have engulfed as well, because remember, plant cells also have mitochondria, all right? But they would have engulfed some sort of a photosynthetic bacterium, some green algae, or some blue-green type of algae. Now, I know I said the other day that green algae refers to plants. It does, but there's a line between when they are plants and when they aren't plants. But nonetheless, all right, folks. So they engulf some sort of a photosynthetic bacterium. And the advantage then is you can photosynthesize. We talked about that. And then we go through a bunch of other steps, and we get, the one with the mitochondria is the ancestor of all heterotrophic eukaryotic cells, which we are. And then the other one is the photosynthetic eukaryotic cells, right? This is called, by the way, the endosymbiotic theory. This is a theory, an accepted doctrine, scientific type. There we go. That explains a few observations that we see in mitochondria and chloroplasts. Remember, that's why we have scientific theories and doctrines to explain observations. And you might have heard this in Bio 1. Hopefully you did. But the observations we had is when we look at chloroplasts today and look at mitochondria today in your cells, well, you don't have chloroplasts, but if you did, we find three very interesting things. Chloroplasts and mitochondria have their own DNA. As a matter of fact, mitochondrial DNA, the DNA in your mitochondria, is only from your mother. It's maternal. It's completely maternal DNA. Isn't that neat? So it's kind of cool for tracing genetics. So no other organelles have DNA. Endoplasmic reticulum and lysosomes and all them other things, they don't have DNA. Number two, the shape of chloroplasts. They're kind of rod shaped. They're kind of bacilli shaped, aren't they? Rod shaped, like the ones we talked about in lab and you looked at in lab. And also, mitochondria and chloroplasts, they can divide independently of the rest of the cell. So the cell doesn't have to divide, but like, for example, if a plant was growing and suddenly the leaves of that plant broke through into this big sunlight, what you might expect to find happen is inside the cells of that plant, man, going to start cranking up chloroplast, aren't they? Whoa, son, man, let's get a bunch of chloroplasts, and so the chloroplast can divide. Mitochondria, you might see, like, say, long distance runners. Folks who do endurance in general, or any sort of heavy physical activity, you would expect to see more mitochondria because you got more energy demand, right? So let's say, and they divide just the same way that bacterial cells do, or prokaryotes do, binary fission. So division by binary fission. All right, so those three observations that we make on today, these things kind of go, wow, that's weird. So this is an idea. Well, maybe these things were not an idea. Remember, in science, a hypothesis is more of an idea. Theory is more of a bunch of ideas that have been supported by evidence. So this is beyond an idea, and those explain those things: the shape, the binary fission, the dividing that we see, and then the fact that they have their own DNA. How many of y'all learned that in Bio 1? How many of y'all learned that in Bio 1 and did you guys discuss the endosymbiotic theory in Bio 1 at all? Give me a thumbs up or anything. Nobody? I must be the only one, though. I think that's so cool. Oh, you did? Yeah, I figured you did. But yeah, it's pretty common, right? Rest of you? Yes, no? Yeah, maybe not. I don't go into that much detail. I just kind of say, yeah, I kind of mentioned the three observations. Yeah. I think that's neat, though. Not only explains, the sophistication that we start to see in eukaryotic cells, how they were able to do it. For one thing, there might have been other things they engulfed, but nonetheless, it's a good explanation for why we have these really interesting observations about those organelles. All right, so what do we got going on at this time? So about 500,000 years ago, give or take a few million years, give or take somewhere between 25 and 100 million years, to be honest with you, this thing called the Cambrian explosion happened, right? So if you're looking down below at the timeline, all these weird words that you see, the Paleozoic and the Proteozoic, those are time periods grounded in geological events, to be honest with you, which is fine because geology is environment, and we know they're all linked together. But somewhere around where you see right there, right, something called the Cambrian explosion. Again, I put a piece of dynamite to say it had a pretty long fuse, and it was an explosion not in space. It wasn't like it wasn't an explosion. What it was a rapid diversification among many different kinds of organisms, and specifically, or most notably, among the animals. So take a look at where that line goes up. Before the Cambrian, I'm going to erase this. Before the Cambrian, we had sponges. We had sponges are animals, believe it or not. We had jellyfish. Those are the cnidarians. Up at the top, the sea is silent. Hydra. We had those. Those were around. Those are pretty simple. We had a few mollusks. We had some snails. We had some worms, annelids. There were no backboned critters around. See the chordates up there? Those were the backbone. And there wasn't any of our really close relatives, believe it or not. I'm giving the story away, doggone it. We're actually very closely related to sea stars, you and I. So anyway, at the Cambrian time, you notice what happened is we go from just a few species. There was a really, I'm going to use the word we used the other day. How cool when we can use these words and remember and know what they mean. During the Cambrian explosion, an extensive cladogenic event occurred, especially among the animals. Cladogenic. So that's the word I used, cladogenic. Remember, cladogenesis is the quick appearance of organisms, as opposed to anagenesis. Anagenesis was the gradualism that Darwin described. They both happen, right? They both happen. Organisms can evolve either way. But when you see this branching in a relatively short period of time, again, 25 to 100 million years, right? You see this rapid of branches coming out of the tree. You go, that's an explosion in diversity. See, remember when I said that the Cambrian explosion bothered Darwin? Because the Cambrian explosion is you're looking through fossils, you're looking through rocks that are that old, and you find there's a few critters, and all of a sudden in newer rocks that were laid down, within a period of about 25 to 100 million years, you get a ton. Boom! You're like, whoa! That bothered Darwin because he's like, that's a lot of time, 100 million years. Earth's a lot older than 6,000 years, but that's still kind of fast. So he really wasn't comfortable with that. It wasn't until the 70s, 1970s, right, that we figured out this idea of punctuated equilibrium. So there was this massively cladogenic event. Boom, we went from a few organisms to a whole bunch of organisms, the Cambrian explosion. What drove this? An arms race again, folks. At this time, larger critters, just like it did back maybe when the engulfing occurred, right, developed the ability to eat smaller ones. And predation, like I said before, exerts a very heavy selective pressure. Or to be really much more accurate, since I don't want you to think about evolution as selecting against, I should say evolution of predation really selected for an advantageous variation. And that would have been something that you could avoid predation. Armor coating, being really fast, only coming out at night, being meaner than the predator, being bigger than the predator. A lot of herbivores did that. A lot of big herbivore dinosaurs, they said, the heck with fighting you. I'm just going to be big and have these armor things all over me, and you're going to have just to beat the crud out of yourself to even eat me. So, okay? See what I mean by the Cambrian explosion? See what I mean by explosion? Not really an explosion, right? Just a rap. And this happened in other groups too, not just animals, but animals very notably. Okay, so we're going to kind of run through this stuff. As I said the other day, when we get into different groups, I'll look at their evolution a little bit more. But right after the Cambrian explosion, we get this movement out onto land. See, this Cambrian explosion thing that's happening is happening mostly with organisms, aquatic organisms. Moving on to land hadn't really happened yet. Like maybe some algae and some bacteria, but really getting out, and especially fancier plants, especially plants, not just mosses, who might have been clinging on at the edge of the water. Soon after this Cambrian explosion, relatively soon, we had organisms that began to move out on the land. Now, first, that would have had to have been the plants, right? If you just think about that without even really thinking scientifically, doesn't that make sense? That the plants would have colonized the land. Don't worry about the word mycorrhizae is. I'll talk about that in a second if you're looking at the slide. But doesn't it make sense the plants would colonize land first, huh? I mean, come on. They are the producers in any ecosystem. They are the critters that everybody else needs and depends on. They're the glue that holds it together. No plants, no herbivores. No herbivores, no carnivores, etc., 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 right? So to move out on the land, the animals would have had to follow the plants. And so the plants colonized land. And soon after that, then animals. And what they were mostly, at least initially at about 450 million years ago, were the arthropods, the insects, the critters that, as we'll learn a little bit later on, come to find out have these very unique adaptations for living on land, very unique adaptations. Ones that would not have worked in water at all, would have actually been detrimental. So it's really kind of neat to think, mind-blowing sometimes, to think about the mutations and the changes that occurred to end up with this kind of diversity. You see, one of the biggest things when you move onto land is the fact you're going to dry up. Right? Again, that's without even thinking about science. What's the biggest thing? Once organisms in the water, moved out on the land, they had to think about, oh gosh, I'm no longer in water. I'm no longer always moist, which is important for life. We got to be, things move across membranes and cell cytoplasms. Cells are mostly water. That whole thing we were talking about in class the other day about enzymes and substrate getting together. So most of the animals before the land plants were like water. Yeah. Now, remember sometimes what we talk about before, it's just a little bit before. But yeah, the initially organisms, animals, herbivorous, I almost made a mistake there. There's no such thing as a photosynthetic animal. Herbivorous or animals would have needed land plants, absolutely. was talking about drying out, right? Desiccation, yeah. So, that's a big problem with organisms moving out into a new environment is how do we prevent from drying out? In the ocean, that's not an issue. But again, the enzyme and the substrate get together by randomly bumping into one another. Now, I was kind of doing that little silly exercise in class where I was walking around bumping into the walls. Remember, that happens a lot easier or a lot more readily if those things that need to bump into one another are dissolved. They're liquid. They're in a liquid state. The cytoplasm is a liquid state. Holy cow, the earth is a liquid state if you think about how much water there is. Again, back to Bio 1. One of the properties of water, solubility. The ability to dissolve things, because water in the cytoplasm of the cell is like the beer cooler at a party. It's the social lubricant. Everybody wants to mix around it, right? Everybody is going to mix and mix and mix in that kind of an environment. So that's what water does, right? And now when you move out on the land, you got that problem. You ain't got water. Now you can use air, and we'll talk about a little later on how that works in some critters, but at least initially that wasn't really an option. So plants that moved out on the land, one of the things they had to deal with is their leaves drying out. Now, their leaves they needed for photosynthesis. Oh, gosh, it's like this thing in evolution that's always, for one, take away one, or give 3, take away 2, or something like that. For every advantage, like big leaves. Think about that. If you moved out on the land and you developed big leaves, and not all plants have leaves, moss doesn't have leaves, but if you develop leaves and then bigger and bigger and bigger and bigger leaves, you're like solar panels, man. You could suck in a lot of sunlight, but now there's a problem. Big leaves heat up. They absorb a lot of heat. And in the air, as opposed to the water, you're going to heat up faster. I know what I like to do at Siesta Key every year when my wife and my family go down vacationing. I like to float in the water because I don't heat up, right? That's why we go to the beach. All right. So now this is interesting because we're going to see environment is an equal opportunity pressure. In other words, animals, as we'll talk about later on, also had this problem when they moved out on the land. How do they prevent from drying out? And by the way, the insects were one of those that figured out how to do that 450 million years ago. Okay, now, plants moved out on the land, right? And they started creating opportunities for everything else. Not only for things for animals to eat, but another thing that plants tend to do is they change the environment. They change the chemical makeup of the soil. They incorporate atmospheric nitrogen. There's a good time as any to talk about this. Plants need, typically in plant growth, we said the biggest limiting factor to plant growth is the amount of nitrogen in the soil. Now, if this seems a little weird to you, I hope it does in some degree, unless you know about this already. You know that most of our air is nitrogen. Like 70% or something? So why do we say nitrogen is limiting to plants? Well, because I said nitrogen in the soil. You see, plants can't absorb or use atmospheric nitrogen. They need nitrogen that's been turned into certain chemical compounds, nitrates and nitrites. And one of the things that does that are prokaryotic organisms. And those prokaryotic organisms oftentimes, or in many cases, will live in conjunction with the plants. Like, for example, if you pull up a bean plant, legumes, beans, peas, all that sort of stuff, you might see on the roots, there we go. You might see on the roots these little nodules, lumps. Not if you're picking potatoes. I'm not talking about the potato, but they're a little smaller than that. Those lumps, those little nodules, are full of nitrogen-fixing bacteria. What we mean by nitrogen-fixing is those nodules are full of bacteria that take atmospheric nitrogen and fix it. By fix it, we mean do something to it chemically to incorporate it into another compound, like a nitrite or a nitrate. That's what the plants can use. So not only do plants provide food, they started providing opportunities for other plants because guess what? Those bacteria that live with those plants, they produce a lot of nitrates and nitrites. That's fertilizer, folks. They produce a lot of fertilizer. when you go to the store and you buy fertilizer for your plants, you'll notice that there are many times on the bag of fertilizer, there'll be a number, 3 numbers, 15, 30, 5. That's the NP, hold on, K content. The nitrogen, the phosphorus, and the potassium content. The nitrogen, right? So plants didn't use all that nitrogen themselves that had all these nodules of bacteria. So guess what? The soil became richer, became more concentrated fertilizer. It wasn't just barren rock. It was now barren rock that had nutrients that provided for plants, and other plants that moved out of the land were dying and they were decomposing, and now we're getting an organic base in there. We're getting carbon. See what I mean? So, like, basically, they're kind of like giving their lives for each other. You know, they moved out on the land. Oh, my God, we struggle. And maybe it died off and went extinct, but you changed the soil enough for somebody, other organism behind you to get a foothold. Or maybe you're still there. You might have changed the soil, but you're fine. And this other plant says, hey, if you don't mind, can I have the extra? Yeah, that's cool. And guess what? We're all living together. Did I lose anybody? We making sense there? We understand what I mean by plants doing all that? I love this story. I think this is so cool. Speaking of things working together, up here at the top, back to this really cool word, mycorrhizae. Mycorrhizae. What are mycorrhizae? Well, let me show you what they are first. Okay, let me tell you what you're looking at. You're looking at the root of a plant. And you're looking at the root of a plant in a cross section. So in microscopes, what we mean by this is the root. What they've done is they've chopped a piece through like this, thinner than that, obviously, and they've laid it down on a microscope slide. So you're looking through it this way, right? So this is the interior of the root of the plant. Those are the cells, the conducting tissues. Now, what is all this around it? Around the outside of the root, in this rim right here, and extending down into this area, your darker circle, are fungus. It's not a bad thing. Fungus isn't always a bad thing. But what we mean by opuscule containing cells is an opuscule is something that looks like like a bunch of grapes, kind of. Now, what that is the fungus that's growing in amongst the cells of the plant's roots. So those cells that you see there, that you're looking cross-section through, in between all of them, there are all these fungal cells. Now, you might think, well, like I get fungus, that's a bad thing. No. They're in a symbiotic relationship. See, what fungus do that we'll learn about a little bit later on, fungus are really bizarre, is they digest their food on the outside. Like, okay, we digest our food on the inside. If you're trying to think, why is he emphasizing that? We bring our food into our stomach and digest it there. A fungus, what it does is, and we have enzymes in our body that help us in digestion. A fungus digests, secretes those enzymes into the environment through all these little, they're not roots, but we'll learn what they are later on. So all these little tendril-like things, and it secretes all these things called exozymes, enzymes that are used on the outside. And they digest all the organic material in the environment, and then they soup it up. They suck it up, right? What a cool way to do things. Now, here's the nice thing for plants. When those fungus are doing that, it's kind of like the roots on the nodules, right, on the legumes. The fungus dissolve more nutrients than they need. So the plants get the extra. And you're already growing in a difficult environment. You're some of the first plants out on land. So this may just provide enough of an edge for you to survive. So the fungus gets something from the plants, and we'll talk more when we get into plants, but, you know, the fungus gets basically carbon material from the plant so it can build its structure, and the fungus in turn helps the plants by digesting stuff. So the plant roots are in this, you know, nitrogen-fixing bacteria, digesting, you know, rich soil, right? And again, it doesn't have to be perfect, but just enough to give you an advantage. Now, mycorrhizae, the fungus, that's what the name of the fungus, the darker ring that you see there, are so important that even today, 90% of all plants that we consider vascular We'll talk more about plants and what that means. But vascular plants have these conducting cells, these tubes in them to move water and things around. 90% of all vascular plants have mycorrhizae on their roots. 90% on Earth. So it appears that as a plant, with few exception, if you're going to live on land, you got to have these mycorrhizae. You got to have these symbiotic. So in some cases, symbiotic relationships are, yeah, cool if you can get them. In some cases, they're absolutely crucial. All right, any questions on any of that? Isn't that neat? I mean, you just look at simple things like that. Do you realize, you know, the fact that 90% of all vascular plants, most everything you eat that's a plant is vascular, makes the difference perhaps between us even being here? Because if there hadn't been mycorrhizae, a few plants might have moved on the land, and who knows? Who knows what would have been in the evolution of the Earth? That's what I think is so cool about this. We're discussing how our own existence came about. And so interesting to me how many times along the way, you know, we dodged a bullet. And when I say we, I'm talking about this collective diversity of life on the planet, because throughout history, we can recognize a number of extinctions. So that's in the red line, but we basically, we call it the big five. And those are the ones in the red arrows. Back in the Paleozoic, there were two of them, about 450 or 440 million years ago, and one a little bit sooner. I'm not going to discuss those because I want to discuss two of the bigger ones and the significance they had, because they do a really good job of putting together what was going on Earth environment-wise and how that changed the Earth. I'm going to talk about the one that occurred about 250 million years ago, and the one that was about 65.5 million years ago. That's the Permian on the left, or the one at 250. I'm not going to talk about one with 200. And then 65.5 million years ago. You notice that the letter COSD under the period, that's Carboniferous, Ordovician, Silurian. So P is Permian, then T is Triassic, J is the movie, Jurassic, then C is Cretaceous. Mesozoic, always think dinosaurs. Mesozoic is the dinosaur era. All right, so let's look at what I said, the Permian. Let's look at the Permian. So about 240, 250 million years ago, we had this extinction event. Now, how do we know that? We look through the fossil record. It's kind of opposite of the Cambrian explosion, right? You're like, wow, look at all these critters. Look at all, oh, shoot, almost knocked my glass over. Look at all these critters. Look at all these critters. Oh, man, where'd everybody go? I mean, there's some, but a whole bunch of them are dead. As a matter of fact, in the Permian extinction, 90% of all marine species went extinct, were extinguished, were gone. 90%. Not just fish either, marine species. So think about the diversity. Think about this. Think about the diversity of marine organisms today. If you've ever done any snorkeling or scuba diving down in the Keys, think about how many different kinds of just fish you saw, right? And then keep in mind that that's just the remnants of the 10% of species, and not just in the fish, like I said, even less than that died off about 250 million years ago. Diversity is inevitable. It's inevitable, and Mother Nature demands it, and it just the whole driving force behind what's going on. About a third of the insect orders went extinct pretty quick too. That's the thing with extinction events like this; they happen pretty quick. You know, in this case, about 5 million years, which is fast again, folks. When we're talking about many time extinctions too, like I was showing you, form the boundaries between these major eras. So the Paleozoic and the Mesozoic would have been the ancient time going out, and the dinosaurs are getting ready to move in. So the dinosaurs begin to show up somewhere around 240, 250 million years ago. All right, we'll talk about them in just a second, but let's talk about what led perhaps to them and other things that changed. Let's talk about what's going on in the environment. Let's go back to what geology shows us. All right, now I'm sure you guys have seen this at some time in your life, but the Earth is actually like a broken egg. It's a bunch of crustal plates floating on this semi-liquid mantle, and these big, huge plates bump into one another. And there are continental plates and oceanic plates, and plates rise over top of one another, and sometimes they bump into each other and they go up, and sometimes they bump each other and they go down. They form trenches, they form mountains. And sometimes they slip past each other, but they get stuck because guess what? They're made of rock, and then they push and they keep pushing, and all of a sudden they slip, and it's an earthquake. All right, so it's a very active planet. It's what we call a tectonically active planet. And we know the way the Earth is splitting, basically. Or, I'm not splitting. Earth is not splitting. But the way the crust is moving. Crust is being formed along the Atlantic. These are formed. What's it mean formed? Well, it's bubbling up from below. That liquid rock mantle is bubbling up, cooling off, solidifying. And Iceland, for example. Where's Iceland? Right around way up here, right? Iceland is a collection of volcanic material that's just, laid on the ocean floor, then more and more and more until it finally broke through and became an island. That was a really cool video when I was a kid. Oh gosh, was it like 6th grade, 7th grade? The teacher showed where they have video of this island forming in the middle of the Atlantic. I think it was the Atlantic. Anyway, there's this volcano erupting, blah, 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 and all this stuff is. And I think I read the other day that island is still around, and so it was really kind of cool because from biology, it's like, whoa, man, this is kind of like what happened on the early Earth, right? How areas that weren't colonized become colonized, and it did become colonized, right? Critters moved out there, plants and animals and stuff. All right. So anyway, what am I back to? If we can backtrack where these continents are today, this is kind of where they are today. So Atlantic is getting narrower. Pacific is getting, Atlantic is getting wider. Pacific is getting narrower. India is ramming up into the Eurasian plate. This is really cool. India came from all the way down here, sort of like an end run, all the way down here, and slammed in the Eurasian plate, and really slammed hard, and is pushing even harder. That's the Himalayas right there, folks. That's why the Himalayas are the way they are. India and the Eurasian plate are pushing against each other. Think about the forces. Not very fast. I think Mount Everest is getting higher by an inch or something like that every year. All right, so what we could do is we can go back and we can take a look, and we say at about 250 million years ago, when this Permian event happened, what we end up getting is this collection of land masses. So all that volcanic stuff that I was talking about with Iceland, imagine that in a global scale. So at about 250 million years ago, this had occurred to a point where we get this huge continent called Pangaea, this supercontinent called Pangaea. And because of the formation of Pangaea, we get this massive extinction. And I'll get to that in the next slide for just a second, but let's follow what Pangaea does. Pangaea, after a while, begins to separate. And about halfway through the dinosaurs, we start to see at least the northern and the southern continents. There's India right there. Sorry, didn't draw it exactly right. But then you go like about the dinosaurs, the continents were pretty much the way they look now, and then to present day, right? So everything's moving that way. But back to the Permian again. What was it about the formation of these land masses that caused this big extinction? Let's go to a zoomed in picture of the Permian. So there's the Permian, and overlaid are the modern day continents. But the green part is where so there was this one basically big gigantic ocean, kind of an inland ocean. There was the Paleotethys Ocean, the Tethys Ocean, and the Panthalasic Ocean, right? The Panthalasic was going to become the Pacific, right? Okay, now, what did this do to the environment? Well, when these continents began to form, before this it was a bunch of little tiny islands of land, and they grew and they grew and they grew till they grew together. So when we finally get this big Pangaea mass, what we get is a radical change in the environments. All right, first of all, oceans deepen, all right? So we get much deeper oceans. Inshore water area got reduced. Now, sometimes folks have this misidea that the oceans are just teeming with life, and if you're out in the middle of the ocean, it's just like being at a zoo, there's life everywhere, but that's not really true. Most life in the ocean has to occur near land. If you think about it, it makes sense again. You got to have things, you got to have producers. You got to have photosynthesizers. Ain't no plants growing 2 miles down. There's no light. Plants have to grow in shallower water. So if the plants are there, then the things that eat the plants are there, and the things that eat the things that eat the plants, right? So inland waters, inshore waters, that's why Florida is so rich in its inshore waters, are great. Now, also, another thing is a lot of nutrients, necessary nutrients, are running off the land into the water, and they get used up, obviously, or they're more concentrated closer into shore that you are. So having a lot of little islands meant a lot of really rich ecosystems. Okay, a lot of things for organisms to survive and thrive and reproduce and compete and take advantage of Darwin's theory there, right? Of evolution. So you get a lot of species, a lot of different diversity there, because you got a lot of different microhabitats. Just because you're an island, Rhode Island is not the same as the Hawaiian Islands. They both got the word island in them, but they're very different kinds of islands. So islands have different environments. Now they grow together in Pangea. Guess what we get now? We get less inshore water. We get less area. It's kind of like that surface area to volume ratio thing you learned about in Bio 1. But when these islands begin to grow together, if you add up, it's a bigger continent, but the amount of coastal area is disappearing. See, these little islands are growing together, so those inland water areas are becoming less, so there's less environments. And again, that's causing massive species extinction. Not only that, but when these continents form, now you change currents, the way currents flow. And that's really important for many living organisms. Not only where currents flow, but when they flow there. So you're blocking off currents, you're redirecting currents, you're changing the amount of oxygen, you're changing the environment. And you're not changing the environment for good in this particular case, especially among these organisms. But remember, one man's poison is another man's food. So the extinction that's going on in the Permian allowed something else to happen. Just like with the plants. What happened? Well, when you start forming land, the bigger the land masses get, because of the way the climate works, the interior of the land masses become drier. So you get what we have now, coastal wet areas, inland deserts. Didn't have that when you were on the island so much. It's all has to do with the amount of rainwater that gets absorbed. Again, it's meteorology, it's climate. But what we do here is we ended up creating another set of habitats. We came up with the dry habitats, the semi-dry habitats, the dry and wet habitats, the dry and dry habitats, the cold dry habitats, and the hot dry habitats. Whatever they were dry. That's the yellow that's in the picture. Now, where do you find a camel? Remember I told you, well, did I tell you all sometimes as a teacher you do hear dumb questions. Is the camel a desert animal? Where do you find it? You find it in the desert, don't you? And the reason why you find it there is because it's adapted to live there, right? It wouldn't do well in areas like the tundra where it's really cold. And by the way, very dry. Tundra is very dry. Just because you see snow doesn't mean it's snowing all the time. It's colder than heck, but it's also very dry. So we get different types of climates, everything in between, by the way. Now, there is one animal out there. Now, there are plants, but there is one animal out there, even today, that's really good at living in drier climates. Not so much the colder ones, but certainly the drier ones. We'll talk about those in just a couple of slides, because there was something else going on during this time, and it kind of hallmarks back to the beginning. The Earth was belching fire. This volcanic activity that kept life, kind of at bay at the beginning, reared its ugly head again, not quite to the level, but we get this extensive amount of volcanic activity. I'm sure if you looked at the geology books, there's a reason behind it. But when I'm talking about extensive, I like the map down below. It's all in Russian. Isn't that cool? It's the only one I could find, although I have found one in English since, but I'm going to keep the Russian one. The area in the blue squiggly line with the purple and the red, indicates what we call a lava caldera. It's a pit of fire, right? Like in the picture in the upper left. It's a massive, huge lava lake, volcanic, nasty, must look like right on the hinges of hell going into it, right? And look at the size of this. It's what covers a third of the country of Russia, modern day Russia. So we're talking about not just a few volcanoes going off here and there. And of course, remember, volcanoes dump a bunch of stuff in the air. And of course, that causes the sunlight and all the debris they dump and all those things we talked about early on. Not so much the reducing atmosphere, but certainly all the junk that gets dumped up into the air. Plants, for example, in that area would have trouble photosynthesizing. Kind of a precursor what killed the dinosaurs in a little bit. So you see what I mean by the extent of that activity. But anyway, that and the volcanic, or that and the continent coming together at the Permian is when we is what we think led rise to this massive extinction. So we lose what we see 90% of marine species, about a third of the insects we create, though. I remember back to that, we create dry land. We create drier lands. And as I said before, there's a critter today that's even good at surviving in drier environments, and that's the reptiles. Reptiles are good at surviving in dry environments because they're covered in an armor coating. It's not just for protection, right? It could be scales. It could be the things we call scutes on an alligator. It's not just for protection that they have that, although certainly in the case of alligators, that's really beneficial. But it also is because whereas the plants begin to put things on their leaves to prevent them from drying out, plants begin to produce waxier leaves. Animals begin to cover their bodies. Arthropods were doing it with their exoskeleton, by the way, and this was allowing them to survive in drier climates, because when you're covering body, you were limiting your water loss. So these guys were really, really good at it. Oh, by the way, after the Permian extinction, it was like they had the place to themselves. They didn't start off big, by the way. They didn't start off to be giants, but since there wasn't nobody else around, and since it's like when your parents go out of town for the weekend, oh my God, you have a party, right? These guys had the place to themselves. They were exploiting new environments. They were reproducing and diversifying and getting big, and not all of them were big, right? Just the ones we were fascinated by. There were also some really small and pretty deadly ones as well. And some began eating each other, and this whole idea of the jaws thing was really happening here. Tyrannosaurus, the guy in the middle, probably wasn't as vicious as we think. At least in terms of attacking. Some of the stuff I've said that said that Tyrannosaurus was actually a scavenger. It didn't make a lot of the kills itself. It would go up, look at those little arms in the front. I mean, how could you do much with that, right? It would go up and roar with these big teeth, and whatever was standing around the kill would say, whoa, we're out of here, and he would eat his part, and then they come out and eat the rest. So maybe he wasn't quite, All right, so the dinosaurs proliferated, and they spent about 200 million years with the place to themselves, more or less. Now, mammals were around during this time. Remember, it's not Aristotle. Mammals were evolving even back towards the beginning of the dinosaur era. They were evolving, but they were having a rough time of it. These guys were kicking our butts, no doubt about it. And we'll talk more about that later on, but it led a lot to the different kinds of mammals. At least at the very beginning. It explains what we see in mammals in the fossil record from that time. But it all has to come to an end. And one day the dinosaurs, while they were enjoying the place to themselves, a big rock about the size of Manhattan, by the way, about the size of Manhattan, came screaming down out of the earth, moving at some incredibly obnoxious speed, smacked into the earth, hitting it, I'll come back to this in a second, hitting it just about where the Yucatan Peninsula is today. All right, so this is 65 million years ago. So where are the continents 65 million years ago? So we're right about here. So right about there. Man, good thing we weren't around. Right about there, that little blue dot that I put on there, 65 million years ago, that crater or that asteroid slams into the Earth. Oh my God, I've seen these incredible computer reenactments, like the Earth, like the crust of the Earth just peels away like an orange. Think about that little meteorite that hit over in Russia, and that was only like a couple hundred feet. This was the size of Manhattan. So this asteroid smacks and hits the Earth just near Yucatan, the Chicxulub crater. They discovered this through oil exploration. They were actually mapping the sea level or the sea floor off the Yucatan Peninsula in this area. And they discovered, when they looked at the results, what looked like a big crater that was part underwater and part on land. So here comes, there they are enjoying themselves. This big rock comes out of the Earth and hits the Earth, and it's what we call a planet killer. In a macabre way, NASA scientists and folks who track asteroids have sort of weird names for them based on size, like town killer, country killer, planet killer. You know, this was a planet killer. This one had the ability to totally change the face of the planet, and it did. Killed off the marine species again, man. I tell you, they got hammered every time. Marine species, especially those living near shore, are going to be very affected by what happens on shore. Not so much the ones out there in the land or out in the middle of the ocean, though. A lot of families of terrestrial plants and animals, and all the dinosaurs went extinct. Now, interesting idea about how we figured out it was an asteroid. When I was a kid growing up, it was always, what happened to the dinosaurs? Why did the dinosaurs go extinct? And there was a lot of theories out there. So it was sort of fascinating to me when this Alvarez theory came around. And the Alvarez theory came around based on an observation. The observation I've highlighted in the picture there with the red arrow is pointing, it's a little bit below it, actually, I'm sorry, that black line. Everybody see that black line, that layer of rocks? So obviously, look at the layer of rocks. Like Lamarck, those layer of rocks are forming the newer layers at the top. And if we know how to date the rocks, we can know how old they are. Well, the rocks before the black line are before the dinosaurs went extinct, so 65 million years. And the rocks afterwards are after 65 million years. Now, notice how thin that black line is. So whatever happened during this time, you know, and we know the dinosaurs went extinct because we find dinosaurs here, no dinosaurs here. So whatever happened during that time when that black line was forming, that rock layer there, caused some major event, happened pretty quick, and killed off the dinosaurs. So this is the cool little detective part. Alvarez is named father and son, I think. University of California, Berkeley, I believe, as well. So when they examine what that black line is made up of, when they find out what the element's in there, they find this element. They find the element iridium. It's probably an element you haven't heard about. You may have talked about it in chemistry. If you wear Oakleys, I don't know if anybody wears Oakleys anymore. Oakleys used to advertise that their lenses were coated in iridium because it would do something. Who knows? Anyway, iridium is a very rare element on Earth. Very rare. But it's very, very common in asteroids. Okay? Isn't that cool? So where did that little black line of heavy iridium come from? And it's all over the Earth, by the way, this black line. You go 65 million years ago in Europe, Asia, and the United States, and you dig down that, you're going to find that black line. That is the remnants of that asteroid. Big asteroid hitting the Earth and boom, exploding up into the Earth, knocking a bunch of Earth rocks, but also itself exploding, and all this iridium raining back down on the Earth and collecting in like a layer of dust on your furniture all around the planet. Now that tells you right away how big this doggone rock was, right? So no surprise that it caused these extinctions. And the dinosaurs, they went extinct. One of the biggest reasons, if you didn't get killed by the tidal wave or because here's what happens when the chunks of the Earth rocks go back up into space. When they start coming down, they gain friction, and you start getting pelted by these flaming meteorites everywhere, man. Couldn't have been much fun. But even if you survived all that, the sun was getting blocked out by all this crud. So the herbivores started to die off, and the dinosaurs. Anyway, it was bad. And the dinosaurs went extinct. The mammals survived, though, and other reptiles survived, too. Reptiles fish didn't go extinct. But the mammals also survived. They were the small little critters again, and they because of the lifestyles they led, because the dinosaurs went extinct, to be honest with you, they were able to rise to prominence after that. But we'll talk more about that later. But what we saw in general among critters is this adaptive radiations. So right after about the dinosaurs, so right about here, 65 million years ago, we see this just like it's a trumpet. We see this rapid diversification of placental mammals, relatively rapid diversification of those with marsupium, and then the monotremes that we'll talk about. Monotremes are very odd mammals, platypus. Not much happened to them. Well, nothing really happened to them. But we see that not only in animals. After this period, after everything cooled down and the Earth kind of healed itself, we see, for example, we look at the Hawaiian Islands. We see that there were these regional events of adaptive radiation. The way to follow this is Kauai 5.1 million years ago. We have evidence of this species, and then as you move down the Hawaiian Islands, you see organisms that are related to that species but show up later on in the islands. Now, what this doesn't show you is the Hawaiian Islands are an interesting study in earthquakes and earth movement too, because the way the islands are forming is because a volcano or a soft spot in the mantle below, and the crust of the earth moves over top of that, moving off to the northwest. And every time it moves over that soft spot, land material bubbles up as a volcano. So the Hawaiian Island is the newest island. But all kinds of regional and worldwide adaptations occurred among organisms after the Cretaceous. Like I said, it was a big time for all. I've eliminated some stuff from your slides that you might download online. I decided to do that. I'm taking that stuff out of the lecture, so don't worry about that. But I do want to talk about this. This is really interesting. And I've also changed the order of things. I'm sorry if that's confusing. But one of the things that we've always wondered about is how do these certain really interesting evolutionary novelties come about? What I mean by evolutionary novelties are things like eyes and legs and feathers. And what we're trying to say here, Stephen Jay Gould, a scientist, a contemporary, not too long ago. I can't remember if he's still alive or not, but he posed a rhetorical question in one of his books where he talks about what good is having 5% of an eye. Now, the number doesn't mean anything, but what Stephen Jay Gould is asking is eyes are pretty complex structures. If you think about your own eye, if you remember from your science classes, right? There's the lens, and the retina, and, there's a lot of, it's like a really intricate structure. So the question has always been, how could such an intricate structure like that arise and be beneficial when it's so complicated? In other words, it's kind of not so parsimonious to think that the eye, the way we know it today, like your eye and my eye, evolved that complex. And if it wasn't evolving that complex, or if it didn't evolve that complex almost instantly, then it was useless. That's what he means by part of an eye, the way we know it today, is not going to be helpful. If you're a bird, when it comes to feathers, part feathers isn't going to be helpful. If we assume that feathers are used for flight, which is very advantageous for birds, right? I know not all birds fly, and we'll talk about that. But feathers, if they can't fly, or what good is 5% of a feather? What good is a flightless feather? What good is a leg that doesn't help you out on land? So how do we explain how these things that have a relatively intricate structure arising when we know evolution, clinogenesis is fast, but it wasn't like one day an organism woke up with a mutation and said, whoa, I got an eye. Didn't work that way, right? Well, we have a word for this. We call them exaptations, the word down below. Exaptations are structures like eyes, legs, and feathers that evolved for one purpose. That was the initial purpose. They evolved for one purpose, but because of what they are, they ended up becoming something fortuitous in the future, in another context. It's kind of like the recessive allele thing again. You couldn't know this was going to be this way, but it did. So let me give you a little hint. We're going to talk more specifically about eyes and legs, but I'll talk about feathers for just a second. We, of course, associate feathers with birds and the ability to fly, and indeed, that's what many birds do with them. Not all birds can fly, though. Some are more flightless than others, right? Chickens versus, say, penguins. But birds are also warm-blooded. And another thing that feathers do is they keep organisms warm. So warm-blooded critters like you and I, we use hair to keep us warm, or in case of humans, we use clothes. We get out of the cold. We hibernate, some of us do. Because from bio one, remember, cold means reactions go slower, so we want to stay warm. Cold-blooded critters, aside for just a second, because birds are warm-blooded critters, and the feathers that initially evolved on them initially evolved for them to survive warmth. To survive in the cold by remaining warm. So these, since birds are really reptiles, these might have been the organisms that figured out how to deal with a dry, cooler climate or cold climate. Right, the feathers initially evolved for warmth, just like a down coat or anything feathers like that keeps you warm, right? Now, that's not a thing for other reptiles other than birds because they're cold-blooded. They don't worry about staying warm. They use other methods, right? Talk more about that in the reptiles. But the birds have these feathers, and of course, what happened through time is these feathers were changed and mutated. And all of a sudden, they became different kinds of feathers. They became feathers that could fly, right? So we'll come back to the feathers part, but let's and the legs, but let's do it with the eyes. Let's kind of follow an example of how 5% of an eye could actually be beneficial. Beneficial, convey an advantageous variation. Okay, let's look at some modern day critters that exist around today, all right, but might give us evidence of what organisms years ago developed in the form of eyes. So, for example, in the upper left, we're looking at an example of a limpet. A limpet is a mollusk. Patella. You have a patella as well. It's your kneecap. But patella is a limpet. It's snails, clams, oysters, limpets fit in that group as well. All right. Patella, a limpet, has an area of pigmented cells, an area of cells that are considered photoreceptors. So what those cells can do is they can sense light. That's it. Just light. Light and dark and shadows. Now, okay. How would it be beneficial to an organism, right, back in the past, or even the ones that are around today, to have a spot of cells that can only tell light? Well, it's not going to be beneficial if they're going to try to read a stop sign. I'm not going to be able to do that. But now, if an organism today or back then had these pigmented cells, and maybe it lived in the ocean, and maybe it had a predator up here that would occasionally swim over top of it, while it couldn't necessarily tell if it was the predator or not, it could always err on the side of caution, and when it ever sensed the shadow moving overhead, it would duck into some grass, say, and hide. Now, keeping what we learned about evolution, do you think having those pigmented cells in order to tell that a shadow is moving overhead, do you think that might convey an advantageous variation to that critter? What do y'all think? So having just simply pigmented cells, do you think that alone? Jesse, how about you? Sure, it might help. Yeah, why couldn't it? I mean, if organisms responded to that shadow moving overhead by ducking into cover, those organisms would survive longer than other ones. And they can't see. And you might say, well, they would duck into cover for any shadow. Well, yeah, but at least some of the time they might be ducking into cover to get away from the shadow of a predator. So now we have an organism, a mollusk, another mollusk, kind of like a limpet that we find around today, the Pleurotomeria. And Pleurotomeria has these pigmented cells, but in a cup, in an eye cup. Now, the eye cup does something very interesting. It gives you a sense of direction. Not only only sensitive to light from above, this thing is sensitive to light from many angles. So you might say there's a sense of peripheral vision. Now that would be, right? That would be also favored by Mother Nature. That would be an advantageous variation. Not only putting the cells in a cup allow you to tell direction, but you ever notice like when it's really sunny outside and if you don't have your sunglasses, you can squint. Or if you're like me in the middle of the night, you want to see what the clock says, but I don't have my contacts in. If I squint a little bit, I can kind of make out the numbers, right? Well, that's what you're doing when you're making an eye cup. You're narrowing the amount of light. You're getting rid of the glare, and you're allowing it to focus a little bit better. So these organisms may have had some or have some very simple focusing techniques. Now, a nautilus is another mollusk. That's the critter that's around today, right? Nautilus is a really cool organism, a chambered nautilus. I'll show you some pictures of them. But in the nautilus's case, we take the eye cup, we add a fluid-filled cavity, we add some pigmented layers of cells to make a retina. So what we're basically doing is running with the same design, but just adding a few little changes every time. And every time, like the pinhole eye with that pigmented layer, that retina, that fluid filled, we start to get a more focused light, and the fluid filled cavity is going to start to end up act like a lens. Because like we look in the next one, the Murex, they have a lens. and a cornea that covers that little tiny eye. And then we go to loligo, which is the modern shrimp. Shrimp have eyes very much like you and I. We have what we call as a camera type eye, camera lens type eye. So shrimp see the world squid see the world pretty much the way we do. Anyway, you see what I mean by this being an acceptation? So back to Stephen Jay Gould's rhetorical question, what good is 5% of an eye? Well, nothing, but didn't start out as an eye. It didn't start out as a lens with a cornea and a retina. It started out as simply cells that can sense light. There are critters around today. We're going to look at one in class, the planarian. When we get to flatworms, planarians have eye spots. And these eye spots are nothing more than pigmented cells. Again, they could just tell shadows. So these kinds of things indicate to us, this is again evidences for evolution. These kinds of critters are able to survive because they can really do well with their relatively simple eye or just a handful of pigmented cells in the environment they're in. Yeah, evolution doesn't create perfect critters, folks, right? It only creates organisms that are good enough at the time. And from a patella limpet's point of view, having just a patch of cells that can tell light and dark works just fine. Right? No goal. Evolution has no goal. It's not like a ladder. Now, I don't expect you to memorize the evolution of the horse, but that is an interesting question, or an interesting story nonetheless. But look at all the other horse-like critters that have existed and sort of the windy path that the evolution has taken. Evolution has no goal because there's no way living organisms can know what the future is going to be like. They can't know it's going to be hit by an asteroid today. The dinosaurs couldn't know that. They couldn't know that the islands were going to form drier climates when the Permian extinction occurred. There is no goal. It's just a random collection of events, and it just ends up, 4.5 billion years later, coming up with all this diversity we see today. Now, don't let this picture intimidate you. I'm not going to give you a question on the exam and expect you to fill in all these numbers and organisms. We're going to go through each one of these, but and the text is kind of small anyway. But I did want you to always kind of keep in mind this connection that's going on right here. This connection between environment and organisms. As the environment is changing now, take away the asteroid from the end of the Cretaceous period, but in so many cases, the environment is causing these changes, these worldwide changes that we see. But overall, what we get on Earth is we get an increasing diversity of organisms, right? We might get a lot of extinctions that occur, but its diversity always seems to be the case. Diversity was rising at the Permian, and then it changed. It was rising in the Cretaceous, mostly among the reptiles, and it changed. So this whole planet we live on is ever changing. I read a neat book many years ago. It's called Zoology After Man, and it basically was what we might expect organisms to look like in the future 50,000 years from now. So it was kind of neat. It's kind of a neat little, what do you call that, brain exercise. Any questions on any of that? Oh. I'd like to get started on chapter 27, at least the beginning, if you guys don't mind. We've only got about another 10 minutes or so, but I do want to start talking about this. Tomorrow I'll continue with chapter 27, and then we can also talk about the exam. I'd like for you all to start looking over the material and have some questions available for me. So tomorrow what I'll do is I'll do 27, set aside some time at the end to go through questions. But I would like everybody to get current on their studying and have any questions for me or things I need to go over again or any of those clarifications, all right? I think that'll be better use of our time. I don't like to do reviews for questions or exams where I basically just sit there and kind of tell you what's. I like to find out where those students are having troubles, because of course that's what we need to talk about most. Okay, now we move on to the part of the semester where we're out evolution, and we're going to start talking about critters. So in chapter 27, we're going to talk about these guys that you see all over the head, or I'm sorry, not the head. This is the point. I think these are neat. This is the point of a straight pin under an electron microscope. The purple is the point of a straight pin. And everything's been color-coded to help you see it. But the yellow You probably recognize those from lab yesterday. Those are all rod-shaped bacteria. I'm all into the idea that prokaryotes are found everywhere. Prokaryotes being bacteria and archaea. They're found everywhere. That's why we do that thing with the plate, which I'm sure when we get back next week will be good and right. But I also think about these pictures when you get a splinter in your finger and you run into the sewing room or wherever, into the junk drawer, and you get a needle out to tear that splinter out of your skin. Remember that the next time you do that, when you tear into your skin, all of the bacteria there that you're potentially inoculating your body with. Although, let's not get freaked out. Most bacteria don't bother us, right? Now, bacteria are really hardy, or let me say prokaryotes. Prokaryotes are very hardy organisms. They can live in some of the most harsh environments on the planet, and this is another interesting piece of evidence when we think about how life, might have began here, as well as how life, or what kinds of signs of life, or what kinds of conditions life can handle. Prokaryotes, for example, there are certain species that are known to be able to handle something like 3 million rads of radiation. A rad is a measurement of unit in radiation, 3,000 times the fatal human dose. They can live in pHs that are so acidic they would normally dissolve metal, in temperatures that are close to boiling. They can survive. A lot of those in those hot springs out in Wyoming and all that. Those things are close to 100 degrees Celsius. And in salinities that are, greater than 7 times greater than the ocean. The ocean is about 3.5% salt. And it's pretty salty if you think about it. Think about spending the day in it. You're always very thirsty when you get out of the ocean. And these guys can live in conditions well beyond that. So when I was talking the other day about panspermia and prokaryotic type organisms being brought on asteroids, you think one of the problems you would have out in space, obviously, would be radiation. And I don't know, being able to withstand 3 million rads might give you just enough of an edge to survive those kinds of conditions. So it's possible that microorganisms or prokaryotic critters could survive a transit in space. This is a neat picture from back the 90s. They found this. This is a meteorite they found in Antarctica. Now, for reasons that geologists could tell you, they determined that the kind of rock this was, or the meteorite this was, must have originated from rocks from Mars. And so imagine a scenario that Mars is hit by a meteor or an asteroid, throws up a bunch of junk into space, lot of rocks and stuff. Most of that falls back down on Mars, but some of it might get high enough up and have enough velocity that it actually kind of goes off into inner space, and then eventually gets trapped by the Earth's gravity and then gets sucked down as a meteorite here on Earth, right? So, that's how the story would have been, and that's not so unusual to think about in terms of geology. But when they started examining these rock under an electron microscope, they began to see figures or shapes or things that were kind of interesting, like the one you see in a picture. This looks a little bit, if you will, like maybe fossilized streptococcus or streptobacilli, maybe rods or circles. So this could is the opportune word here. This could be remnants of prokaryotic organisms living in Martian rocks, life on other planets. Now, I got to be fair with you. There's been a lot of geologists, a lot of prominent geologists that argue that there are geological things, geological processes that happen in rocks that could leave something that looks like this. So it might not be fossilized Martian bacteria. It might be something from the geology, but honestly, we've moved so beyond this now when it comes to the evidence we have for life, that there's a lot more exciting things out there going on, not just on Mars, but on, like I said, some of the moons of Saturn and Jupiter. So prokaryotes, so far we've learned they're pretty doggone hardy, right? They can survive the rigors of space, and they might actually have. So let's talk about what makes them what they are. For one thing, cellularity. They are the epitome of that, right? They're unicellular, most of them. Some come together sometimes and they form things called colonies, and they even sometimes come together in these colonies, or sometimes they're called biofilms, and they get into this idea of division of labor by certain cell types. So certain cell types will handle waste metabolism. Other ones will deal with maybe photosynthesizing, or the case may be. Now, and this is as good a place as any, like the other topic, I want to talk about multicellularity for a second. When we discuss the word multicellularity, there's lots of ways you'll see it called. Multicellularity, in its truest definition, refers to animal tissues. Animal tissues and the multicellularity aspect of them indicate that these are groups of cells with very specialized functions. So when things like prokaryotes form together in colonies, right? Or even when cells in plants form together in larger groups, the real precise technical definition of a tissue is like an animal tissue, like nervous tissue. So just because we have more than one cell living together here, we don't have multicellularity yet. True multicellularity. All right. So when you see that word multicellularity, it sometimes is taken, the context of it can be different, but the real definition of it is the complexity of animal tissues, which we'll talk more when we get into animals. You learned the other day that there's different shapes. There's round ones, rod ones, and spiral ones, or helical. I think I told you DI in class. I should have just said Diplo, but pears, in chains, or in grapes. Obviously, we learned they were pretty small, about 1 to 5 microns. Anabina, the little blue-green ones, are about 5 microns, about 5 to 8 microns, actually. So that's about the average size of a prokaryotic cell, although there are some big ones, relatively speaking. That big one down below, those are 2 pictures, folks. One's a little bigger, zoom in, of Thiomargarita nambiensis. Cool name, huh? Thio means something to do with sulfur. And then it's up against the B to show you kind of a relative size, but it's about 3/4 of a millimeter. So you can see it with the naked eye, but generally prokaryotic cells stay pretty small. They don't get very big. Nearly all of them have a cell wall or a mixture of different types of surface structures. Gloeocapsa had some surface structures that we'll talk about that I mentioned in lab. Sometimes these surface structures can help protect these guys from immune system cells. They kind of hide behind these surface structures. Surface structures can do things like maintain the shape of the cell, again, provide physical protection, or just like in plant cells, they can prevent them from lysing in an environment in which they're hypotonic. Remember, in hypotonic environments, water flows into the cell, right? So animal cells would lyse, they would split in those environments. So animal cells prefer isotonic, while plant cells, they like hypotonic because they have the cell walls, remember? Cell walls, now many bacteria have a cell wall, or many prokaryotes have a cell wall. It's not made of the same materials plant cell walls, not made of cellulose, but it does the same job. Remember, plant cells don't explode when they're in hypotonic environments because even though that central vacuole grows with water pouring into it, it's like blowing a balloon up in a box. It hits the cell wall and you can't blow it up any further. All right, so this is tying back to yesterday looking at Streptococcus and understanding gram-positive and gram-negative. Yesterday we learned the simple part of it. We learned the colors, right? Gram-positive are purple, gram-negative are going to be red in color. All right, let me show you the basis behind this because there's a question in your lab manual. The reason why gram-positive are positive when you stain them and gram-negative are negative when you stain them has to do with something called a peptidoglycan layer. That's what makes up their cell wall. A peptidoglycan layer is a polymer of sugars and proteins, but it makes the cell wall of prokaryotic cells. We don't find these peptidoglycan layers in eukaryotic cells. We only find them in prokaryotic cells. Gram-positive bacteria have a thick peptidoglycan layer, then they got a plasma membrane. Okay? Gram-negative bacteria have a thin, it's like a sandwich. They have a thin peptidoglycan layer. It's sandwiched between 2 membranes, an outer membrane and an inner membrane. Yesterday in lab, I said that one of the reasons why we want to separate these out is because many diseases, bacterial infections and problems in humans are caused by gram-negative bacteria. So this is a very easy way to see if you're dealing with gram-negative or gram-positive. And now, more, on the functional side of it, the reason why is on that outer membrane of gram-negatives, there's many times these little green things look like hairs. Those are lipopolysaccharides, or fat sugar mixes, and those produce endotoxins. Those produce the things that make you sick. Those produce the things that cause the symptoms with these gram-negative bacteria. All right, last slide, then I'll finish for today. Let's talk about how this staining comes about. Because again, gram staining is very important, separating gram positive from negative, and it's due to that peptidoglycan layer. That's why one looks purple and one looks red. So here's the procedure. So we dye everybody with crystal violet stain. Gram positive and gram negative both get purple. We then dump the iodine. Now, the only thing the iodine does is it causes a coalescing of the crystal violet and the iodine. It makes a complex that's found inside the cells. The third step is crucial, and this has to do with the peptidoglycan layer. You wash all the cells with alcohol. Now, alcohol dries things out. So what that does is on the gram positive with the thick peptidoglycan layer, they call it the PG layer here, that thick layer shrinks and traps the cells inside. Okay, the thick layer shrinks and traps, not the cells, but traps the dye inside. The gram negative, theirs is much thinner, so when it shrinks, it doesn't trap the purple dye inside, so the alcohol washes the dye away. Now, you come back at the end and you dye everybody with saffron red. Everybody dyes red, but the gram positive are already purple, so you don't see the red, and the gram negatives get dyed red. You do the red part at the end so you can see the gram negatives too, because they would have no color in them at all until you dyed them. And so this is the basis of gram positive, gram negative, the size of that peptidoglycan layer. Thicker layer in gram positive, thinner in gram negative. Gram positive and gram negative and alcohol both shrink. The thicker one, it shrinks and it's got more density, so it traps the dye, and whereas the gram negative does not. And in the end, they all look like this, right? You can see a bunch of gram positives and gram negatives, and hopefully lots of cocci in theirs, right? The round ones. Okay, let me stop the recording.